Welcome to Lab 2. This week you will work on developing basic microscopy skills that will be essential throughout your study of anatomy and physiology.
By the end of this lab, you will be able to properly care for a microscope, identify the parts and describe their function, explain basic microscope theory, and prepare a wet mount of your cheek cells. Lastly, you will use the microscope to identify cells in various stages of the cell cycle.
Proper handling of the microscope saves time and prevents damage.
Oh no, I dropped it.
Phil, this is why I told you. You must use 2 hands when carrying a microscope. Let's try this again. You must use 2 hands when carrying a microscope. One hand is under the base and the other is on the arm. That looks much better, Phil. When cleaning the lens on the microscope, only use lens paper. Do not use tissues, paper towels, or your shirt to clean the lenses.
Safety and cleanliness aren't just good lab etiquette. They are essential to keeping the microscope in working order and free from damage. Always report any issues you have with your microscope to your lab instructor.
Let's review the main parts of the compound microscope you will use in lab this week. As you learned previously, carry your microscope by holding onto the arm and the base. You turn your microscope on by using the power switch. The ocular lens usually has a magnification of 10 times. The objective lens are on the revolving nose piece and range in magnification from 4 times to 100 times. The stage, with clips, is where your slide will be placed. The microscopes have a mechanical stage, making it easy to move your slide around when viewing. Lastly, we have the light source and focus knobs.
Let's unpack the theory of microscopy. First, total magnification. Total magnification is calculated by multiplying the magnification of the ocular lens by the magnification of the objective lens. The field of view is the observable area of the slide you can see at one time. As you increase magnification, the field of view will decrease. The last part to be aware of is image orientation. The image you see under the microscope is inverted, meaning if you move the slide to the right, the image will move to the left and vice versa. Now we will prepare a simple wet mount of our cheek cells to observe their shapes and nuclei under the microscope. To begin, gently swab the inside of your cheek. Smear this onto a clean slide, add 1 drop of water and a very small drop of methylene blue. Place a cover slip on top and start your observation under low power. Once you find your cheek cells, increase the magnification and draw what you see.
The last activity for this week's lab will have you using your basic microscopy skills to identify cells in the various stages of the cell cycle. You will use prepared slides of the white fish blastula for this activity. Stages to spot include interphase. The nucleus is well defined, the DNA is not yet condensed, and cells appear relatively uniform. Prophase. Chromosomes are visible and the nucleus has disappeared. Metaphase. The chromosomes are aligned along the equator of the cell. Anaphase. The chromosomes are moving toward opposite poles of the cell. And telophase. The chromosomes arrive at the poles, the nucleus reforms, and cytokinesis begins.
We learned about how to properly care for the microscope, reviewed the parts of the microscope, talked about the basics of microscopy theory, prepared a wet mount, and talked about identifying the various stages of the cell cycle.
Remember, when you first put your slide on the microscope, start with the lowest magnification and then work up. Keep the lenses on the microscope clean and slides securely supported. Confirm cell cycle stages by looking for visible chromosomes and their position within the cell.
To finish preparing for lab, be sure to read through the lab in your manual. Once you have read through everything, take the short pre-quiz to assess your preparedness for lab. Remember, the pre-quiz must be completed prior to the start of your assigned lab. See you in lab.